All right, everybody, welcome back. So in our last lecture, we introduced this idea of RNA in addition to DNA. And what we're going to be working through in this lecture is understanding how we get from DNA, which is sort of has the source code for all the protein information that we want, and how the cell is then able to use mRNA to create proteins. So your book has a really great figure that sort of outlines this this whole process of transcription and translation in one helpful figure. And we're going to be going specifically through each of these steps to understand what's happening at each area. And where we're going to be starting is looking specifically now at transcription. So where DNA are transcribed to mRNA, which then exits the nucleus into the cytoplasm and goes to a ribosome to begin translation. And this is where we're really going to begin, is understanding transcription. So it's basically understanding how molecules of messenger RNA, mRNA, are synthesized from the instructions encoded in our genes. So in this case, we have a genetic sequence that codes for a protein area. And we see here the different base pairs. So we have cytosine, cytosine, adenine. And what happens is an mRNA is able to, once the DNA has been unwound by helicase, so the DNA becomes unwound, and in this section, mRNA comes in with the correct coding sequence that matches the DNA region. So where cytosine will pair with guanine, and now we have adenine pairing with that base pair that is unique to RNA uracil. So essentially, mRNA is taking a copy of the genetic code that is written in DNA and taking that code and exiting the nucleus into the cytoplasm where it'll bind to a ribosome. So let's zoom into that process of the DNA unwinding so that mRNA can form. So one of the most important molecules in this process is RNA polymerase. And this important enzyme binds to the regulatory sequence of the gene. Remember how we have a regulatory sequence and a coding sequence. And once it binds there, the gene is unwound, and the DNA, and basically the DNA exposes the coding region of the sequence of the gene. And as the RNA polymerase unzips the gene, the different RNA nucleotides, so these are just free-ranging RNA nucleotides, it essentially synthesizes the RNA nucleotides that match correctly to the complementary strand. And this is how you get this strand of mRNA that is then able to leave the nucleus of the cell and bind to the ribosome for the next step in the process. So we're going to be doing a lot of zooming in and zooming out throughout this lecture, but I thought it would be good to zoom in specifically what's happening. So the RNA polymerase is moving along these DNA nucleotides and is attaching the complementary RNA nucleotides. So you can see just up close what is happening with this process. And the big difference between DNA and RNA, again, is this important base pair uracil, which binds with adenine. And once this mRNA strand is formed, it detaches from the DNA sequence, and then the DNA reforms into its typical DNA helicase shape. Again, and this is because it's going along this, first the regulatory region, then the coding region, and then it detaches and the DNA reforms into that double-stranded helix shape that we're all used to. So once it's complete, we have this completely bound together mRNA. In this case, this is using the example of that antithrombin mRNA from the coding sequence. So again, keep in mind that this mRNA is only the coding sequence, not the regulatory sequence, because we're trying to only take the information that's going to actually encode for a protein. The gene remains part of the chromosome, and then this gene is again ready to be transcribed again to create more protein potentially. So how does this relate to the vaccine candidates that we've been talking about? We know that mRNA, this stretch of a coding region, is what is being used in the vaccine, but what exactly is it? So in this case, scientists were able to analyze a specific protein on the exterior of the coronavirus, and this is called the spike protein, aptly named. And it's very important for binding to host cells. And because we know the code of this protein, we're able to create and manufacture mRNA that would code for this spike protein. So the next step in this process, now that we have this messenger RNA, we're able to leave the nucleus of the cell, is how we get from this coding region of a gene to an actual protein. And that's where we're going to pick up in our next lecture, where we're really going to be diving into translation. Okay, see you soon.